Hello, everyone, and welcome to the webinar titled Mango Malformation and Witches-Broom Disease and Incident in Mangoes. My name is Natalia Tello, and I'm the Research Manager at the National Mango Board, and I'll be the moderator for this webinar. So to start our discussion on Witches-Broom incidents and management in mangoes, allow me to introduce today's speaker, which is Dr. Stanley Freeman. Welcome, Dr. Freeman. So Dr. Freeman is a senior researcher of plant pathology at the Arrow Volcani Institute in Israel. He works primarily with fungal crop disease, which he has done since 1993. He obtained his PhD in plant pathology from the Hebrew University of Jerusalem in Israel in 1991, and his expertise covers plant pathogen interactions of Fusarium, Calotroprum, and other fungal species. He has been working on mango malformation disease for the past 25 years, as well as other mango-related pathogens. He has published over 120 peer-reviewed articles, 30 articles in conference proceedings, 15 book chapters, and 2 edited books. He has also supervised over 30 master's and PhD students, as well as postdoctorate fellows. He is the co-editor-in-chief of the journal Phytoparasitica and has served as the president of the Israeli Pathological Society from 2013 to 2017. So, Dr. Freeman?
Thank you very much for that nice introduction, Natalia. And I'd like to thank you, as well as the National Mango Board, for inviting me to give this seminar. The seminar will be covering mango malformation disease, MMD, and Witches-Broom, and an interaction between them, whereby I will be comparing the etiology, biology, symptomology, and management, mainly of mango malformation disease. Okay, over the years, we've collaborated with many research groups from all over the world, as you can see, from Israel, Egypt, the US, Japan, Spain, Mexico, and from Germany. As far as the distribution of MMD goes, it was first reported in 1891 from India, and thereafter has been detected in the major mango growing areas worldwide. As can be seen here, I'm sure there are other areas regions that have not yet published the appearance of the disease. It's just a schematic world map to show you where it's present. The orange colors indicate publications of where the disease has been detected. As far as disease symptoms go, here you can see typical disease symptoms, which are panicles that resemble cauliflowers. Typical malformation. And obviously, a tree like this is not economically viable in our part of the world anyway. We would not tolerate such an affected grove, and such an orchard would be uprooted and destroyed. Just a close-up of the symptoms of disease. On the left, you have a healthy, as opposed to on the right, infected inflorescence. The inflorescence, which is disease, is mainly fleshy, it's green or red, and which I mentioned before, it resembles cauliflowers. And here you have symptoms of infected Tommy Atkins orchard. Again, the affected inflorescences are very compact and very cauliflowerish. And I'd just like to mention that these types of inflorescence do not bear fruit. And if there are any fruit, the fruitlets drop. So there's a complete loss of yield once you have inflorescence as such. Similar symptoms close up. Sometimes it's very difficult to distinguish between a healthy and a diseased panicle. Here you have wheat inflorescence. On the left, infected, on the right, healthy. And infected buds. Buds, again, also very difficult to distinguish, but usually you find them as swollen. They mainly apical, as you can see here. However, lateral buds are also very commonly found as infected. Here we have on the left infected vegetative malformation. The vegetative malformation is the symptoms are misshapen fleshy terminals with shortened internodes and dwarfed leaves, as opposed to the healthy vegetative growth on the right. Again, another format of vegetative infected as opposed to healthy. We have other diverse symptoms, but all of these symptoms refer to malformed inflorescence. You can have a vegetative as well as malformed panicles together, and this is also very common. Also, the combination of the two. Okay, let's move on to the causal agents. Now, over the years, there have been many hypotheses of what is the causal agent of MMD, and I just go through them briefly: nutritional, physiological, hormonal, viruses, phytoplasmas, mites. However, the red number 6 causal agent, fungi, are indeed the responsible organisms that cause malformation. As far as the different types of fungi that cause malformation, so we have one species or one genus, that's Fusarium. However, with the invent of molecular techniques, we have been able to detect and identify other species of Fusarium that are causal agents. Now, I'd just like to point out that Koch's postulates were first successfully conducted in verifying that Fusarium mangifera is the causal agent of mango malformation disease, and this was conducted in India approximately 60 years ago, later on elsewhere, and in Egypt and Israel, where we published in 1999 for the first time successful conduct of Koch postulates. As far as a species-specific identification using molecular techniques, so Fusarium mangifera is the main causal agent worldwide. There are other Fusarium species, but we are able to detect the pathogen using PCR methods, and how you have a specific single band which will allow us to detect this causal agent specifically. To take it one step further, we use additional molecular sequencing methods to ascertain different species of Fusarium. This is mainly for research purposes, to conduct phylogeny and phylogenetic trees. These are the genes that we use, a combination of multi-locus sequencing. When you use more than 4 or 5 genes, you combine them together, and this way you're able to construct a phylogenetic tree. And this is just an example of the different species that cause malformation. We have different clades. We have an American clade, African clade, and an Asian clade. I'd just like to point out that the Asian clade is that where Fusarium mangifera is situated, and the other clades are much further afield, and it appears, according to the phylogenetic studies, that these other species seem to have jumped from other host plants onto mango to cause mango malformation. Now, all the symptoms are pretty much identical. However, different Fusarium species seem to be involved, However, Mangifera, Fusarium mangifera, which has evolved with mango over the years and originated from India, is a more common pathogen spread out worldwide. So you can see that there are many other different species besides Mangifera. I won't go into them, but again, I'd just like to stress that we associate all these different other species to the same symptomology. But my research, and that has been done in Israel, refers to Fusarium mangifera, which is a very broad worldwide pathogen, and we haven't done any research on any other the pathogens and their epidemiology, because only Mangifera exists in Israel. So this just shows you briefly the different species according to their color. The white you can see is spread out: Florida, Spain, Israel, Egypt, Sudan, China, Oman, India as well. So it's very well distributed worldwide. Okay, now I'd like to now start going in depth into the epidemiology of mango malformation disease, because without understanding how the pathogen survives, spreads, causes disease, we won't be able to create or generate a management or control strategy. So regarding epidemiology of MMD, these are the parameters that we needed to delve into and ascertain to understand how the pathogen causes disease. So we first assessed the incidence when it appears, inoculum availability, when do the spores avail themselves, the patterns of dissemination, how are they spread, and where do they actually infect? What structure of the plant is susceptible to the spore infection? Thereafter, we needed to determine whether the pathogen is localized or whether it infects or affects systemically, and how is it spread? Is it airborne? Is it soil-borne? As such. And taking into consideration all these points, only then would we be able to develop a disease management strategy. So I'll take you through slowly through the different stages of our study on the epidemiology of mango malformation disease. So as far as disease incidence goes, exactly when do malformed panicles form? When do the inflorescence appear in the tree? They appear at different times when normal and healthy inflorescence appear, or do they appear at other times of the mango growth period? So looking at the disease severity, and again, I'd just like to point out we're talking about the northern hemisphere and mainly Mediterranean area. However, you can adapt all this information to your area of the world. And what I point out is that the malformed inflorescences, you can see here the numbers, the tree, and in time according to month, they appear at the same time that healthy panicles appear. In other words, there's an overlap. It's not that malformed panicles will appear after healthy panicles appear or before, but the malformed panicles appear at the same time healthy panicles appear. As far as the inoculum availability is concerned, and quantifying the spores that are spread, this is another very important stage in understanding the epidemiology of disease. So spore traps were set in infected fields using an active trapping method. This is a Burkhardt spore trapper. And as you can see, our period of flowering And again, this is the period when malformed panicles as well as healthy panicles appear. We trap spores in the air at the period when the mature infected inflorescences were present in the tree. And this is an active trapping method using this instrument, which sucks in the air with the spores onto a slide, which you can then quantify the amount of spores that are present in the air. Again, we used a passive method where we placed petri dishes with fusarious selective medium in the orchards themselves. And we saw again there's an overlap between active trapping method and the passive trapping method. In other words, both methods were very reliable and indicating that spores and the peak of spores are released at the period where you have mature malformed panicles. In other words, malformed panicles appear to be releasing spores at the peak of their appearance. Okay, I'll move on now to the availability and quantification of spores. Exactly when do these spores appear, at what time of the month, and when is the peak of the release of these spores. So here you have two consecutive years as well as 2001, and you can see that as the panicles mature, so we have quantitatively higher release of spores. This is a log n conidia per gram quantification method. So you have here 10 to the 5th, so you have even 10 to the 6th. You have 1 million spores per gram of inflorescence. So very, very high amounts of spores are released per gram of inflorescence. We basically weighed the inflorescences and released the spores to see how many spores are available to be spread. So again, you have the peak of spore release and very, very high amounts at the time when mature panicles, infected panicles, are present. Again, this is obviously cultivar dependent, but the cultivars we used, Keat and Tommy Atkins, gave us this picture. Infection sites. Exactly where do the spores manage to land and how do they infect the mango plant? Which structures are susceptible for infection? So we sampled apical buds, lateral buds, branches, and leaves to find out exactly where colonization takes place and what is the most susceptible part of the mango plant that is available to the spores. So as you can see here, the apical buds are the most susceptible structure of the plant. Lateral buds are also very susceptible. Branches and leaves are we barely detected spores within these structures. So apical buds and lateral buds are the susceptible part of the mango tree. Now, another very important aspect which we needed to determine was whether we have a continuous or interrupted colonization of the stems. So we collected many, many infected stems from infected orchards to determine exactly where the fungus is present. Is it present continuously through the stems, or is it localized only at the bud areas, which I mentioned before, either at lateral bud sections or at the terminal buds, or can the fungus grow throughout the the stem, which is a very, very important factor in determining the method of spread of the pathogen and for its control measures. So what we did is we sectioned at the nodes where the buds are located and plated these sections on a fusarium selective medium. And we also plated areas in between the nodes, okay, the internode between the buds, to find out if the fungus is present only at the buds or also at the internode. And in 1-year-old tissue, we found that only nodes were colonized by the fungus, as well as 3-year-old tissues. We found that the buds were the most prominent and most susceptible part that were infected, and we do not find the fungus between the nodes. In other words, only at the buds, at the location of the buds. Looking at infection of seedlings, which is very important due to the fact that you have many nurseries with seedlings which may distribute the disease. So we look, as well as nurseries, at nurseries to see how infection of seedlings can take place. We're mainly looking at vegetative malformation, which is more prominent than floral malformation. And this is just an example of infected mature orchards and young vegetative seedlings growing under these infected trees. And as you can see, these young seedlings are infected. In other words, it appears that the inoculum infected these young seedlings, but the question was whether you have internal infection of the seedlings or only infection at the buds. Again, we used the species-specific primer method, which I'll show you in the next slide how it was used. So basically what we did, we collected hundreds of these seedlings and sectioned them. And as you can see, at the meristem level, we had 100% infection. At the sections in between, we had a much less infection. However, in the roots, we had 100% infection. However, this is plating only on a fusarium selective medium. When we use the PCR specific medium, you can see that the fungus was not root-borne, not within the soil, but only at areas where we found buds located within these seedlings. Okay, just to summarize this intermediate summary till now, we found that malformed panicles serve as a source of inoculum. The inoculum potential highest in May and June. That's when the mature malformed panicles exist. We find that the conidia or the spores are dispersed in the wind, primarily during May and June, and apical buds serve as the primary site of infection. And there is no continuous colonization of tissues, but the fungus is located at the buds only, terminal, meristem, and lateral. There are, however, other alternative inocular sources that we need to check. That was via floral debris. Do seeds manage to transfer the disease? And is the disease systemic or is it only local? So looking at floral debris, this was pretty simple. Looking at the malformed panicles which dried up and fell into a funnel shape of the leaves to see whether the buds were infected. This was set up in a greenhouse. We placed infected panicles within the floral funnels. And what you can see as the infected debris managed to infect the buds at a very high percentage compared to the untreated, non-infected debris. There was no no infection where there was no debris. So debris is a source of infection. Therefore, also this source of inoculum must be removed from the tree. Do seeds manage to harbor the pathogen? So what we did is we went to a healthy orchard and a diseased orchard. You can see that here we have many diseased panicles, but we have fruit as well alongside these diseased trees. Here you have the fruit, diseased panicles. Do the spores manage to infect the fruit and enter into the seed? Again, show you fruit very close to infected panicles. And what we did is we went to trees, we sampled diseased fruit, healthy fruit, and we measured or quantified conidia on the surface of these fruit and found that in diseased fruit there were many, many spores on the surface. As you can see here, the healthy seed as well as the colonized or let's say the healthy seed as well as the diseased seed, none of them were colonized by the fungus. So it appears therefore that none of these seeds were colonized, whether they were surface sterilized, whether they harbored the pathogen or not, but we were not able to detect the fungus within seed. So seed are not a source of infection. As far as airborne nature of the pathogen is concerned, we looked at local as well as systemic nature. Now, this is an orchid which was very heavily infected, and it was cut back and allowed to regrow. And alongside this heavily infected orchid that was cut back, we had a source of inoculum. Here you can see another orchid which was within 5 meters from this experimental orchid. And we want to determine whether the pathogen can be spread by wind into this other orchard. If it was infected systemically, we would have a uniform infection. However, if it was only infected by the source of inoculum from this adjacent orchard, then we would have a gradient of infection according to the spread by wind from the inoculated orchard, from the orchard which was heavily diseased. So here we have a schematic diagram. We have an inoculum source from the adjacent infected orchard. The road in between these two orchards, our experimental plot here, and we have a contour map. And what we can see here is that the orchard, our experimental orchard, which was very close to the inoculum source, was the most infected. In other words, the closer you get to the source of infection, the higher the infection or disease incidence was detected. To map it, to take the contour map and put it on graphic data, here we have malformed panicles and distance from adjacent infected orchard. So as you distance yourself from the infection source, then the infection is reduced. So in other words, the gradient of infection from the source of the infected panicles, and this again determines that we have a gradient and not a uniform infection source, not a systemic source, but a local source. So to summarize the local, as far as systemic nature of infection, there was no infection via seeds or soil. So this determines, or one determination, that we have a non-systemic infection. The production or release of inoculum from infected panicles again indicates we have a localized infection. The pathogen is concentrated at and around apical and lateral buds at the nodes. That determines that we have a non-systemic infection. And again, there's no continuum of colonization within vascular tissues of the stems, again indicating we do not have systemic infection. Now I'd like to move over to the witches-broom symptomology caused by the mango bud mite. There's been controversy over the years and much misidentification of the disease incidence caused by the mango bud mite and confusion between mango malformation disease and witches-broom caused by the bud mite. Just to show you symptoms of the bud mite that causes the witches-broom symptoms, you can see the drying of terminals, stunting of vegetative growth, dieback of the panicles. Here we have distorted growth of the terminals, arrested growth, stunted young stems, and later on leaf fall and drop, and symptoms which are determined as witches-broom, and they are definitely different from mango malformation disease. The phenology of the bud mite, exactly when does it appear as far as the cultivation of mango is concerned? So we find the bud mite just about throughout the year. However, mango malformation disease, as I mentioned, does not appear throughout the year. So this again determines that the bud mite has a different type of symptomology compared to mango malformation disease. As far as the bud mite is considered a major pest, and it occurs just about wherever mango is grown. It proliferates within the terminal buds and throughout the year, but peaks during the spring months mainly. The larvae and adult attack and feed mainly on the terminal bud tissue, causing bud malformation, not floral malformation. Symptoms completely different from malformation disease. And the mites are windborne. They stand erect and are dispersed in this manner. This is the biology and lifestyle of the mango bud mite. Now, how does it interact with malformation disease? Well, before we get into that, I should just stress that the mango bud mite is not directly involved in the appearance of mango malformation disease, which I mentioned before. As an example, mango bud mite is present in Australia. However, mango malformation disease does not appear in that country. This is another indication of the two different symptomologies and two different types of disease. And postulates for mango malformation disease were never proven using the bud mite. In other words, if you inoculate with the bud mite alone, you will never achieve mango malformation disease. What is the interaction between the two? Well, these are roles that we had to ascertain to determine how the bud mite interacts with the fungus. Do they act as carriers, and do they vector the conidia or spores into the apical buds on their bodies? Do the mites enhance infection? And do windborne mites carry conidia on their bodies? So all these questions were answered using our experimental designs. Here we marked strains with fluorescent GFP fluorescent strains. And here you can see the strains of Fusarium with the bud mite. They appeared to cling to the bud mite body, but there was nothing particular about them, but just a mere association. This experiment we set up to determine whether it was possible to vector conidia or spores into the apical buds using the mites, and what we had here were spores and mites in different combinations, and to see whether they moved the spores into the apical bud. And this is just a schematic video showing you the bud mite actually taking the spores into the terminal bud. And did this actually take place? Well, this experiment was set up using different treatments, where we used an untreated control where we never achieved any conidia or no mites within the buds. If we used the conidia alone, again, there were no spores found within the buds. However, if you used mites alone in that experimental design, you found that the mites did arrive at the terminal buds. And if you mixed them together, you were able to achieve infection of the buds, whereby the mites moved the spores into the buds themselves. So they do vector the spores into the buds. Do they enhance infection? So similar experiments were set up where we used conidia alone infecting the buds, and we infected in combination with mites. So this is just plate to show you that conidia or spores alone were infected. So these are buds taken from infected seedlings, plated on media, and you can see that if you put conidia into the plates, into the buds, and put the buds on the plates, there is a degree of infection. You don't need the mite. The conidia will infect by themselves. However, if you have a combination of conidia with the mites, you do get enhanced infection percentage. Here, this summarizes the quantification of the interaction between the conidia and the bad mites. So when you have mites and conidia, you have just about double the amount of infection. Wind-borne transport of conidia, do we find conidia on mites which are placed in the field? So here we have slides placed within this tunnel, rotating in an infected orchard. And what we found was that very few bud mites were caught in the orchard trap, and none bore conidia. In other words, the spread of inoculum is mainly by wind and not by virtue of the mites. So to summarize this part, airborne conidia are disseminated from infected panicles, while buds serve as primary infection sites. The disease incidence is dependent on distance from inoculations, on inoculum source. We found that infections are local and not systemic, and the bud mites, they mainly act as external carriers of conidia. They can vector the conidia into apical buds, enhance infection, but do not appear to bear the conidia and disseminate in between trees. Just summarizes the disease cycle. You have inoculum availability during flowering, and the inoculum is released from the infected flowers. They are dispersed by wind at the peak of inflorescence formation. The inoculum lands on the leaves and buds. They can infect alone the conidia. However, if there's presence of the bud mite, then they can assist the conidia or the spores in reaching the site of infection, the buds, and you get more severe infection with the presence of the mites, and eventually you will have your systematic buds, which the following year will break, and you will get your infected panicle. So this is the summary of the disease cycle of the Fusarium pathogen. Now I'll move on to management of the disease. I'd just like to say that the management is based on the fact that the pathogen is airborne. We have airborne spread of the conidia, and I would like to make a terminology of a specific infection window. I'd like to mention that window of infection is also the window of protection, and I will show you this point in the next slide. Here we have a model, schematic model of the disease epidemiology. These are the months of the year, a 2-year continuous growth in an infective orchid. Here you have your diseased panicles releasing spores, say from April through to July. This is the available inoculum which infects buds. So basically buds which are available to these inocular sources will be available for infection. And we have a latent period where these infected buds will remain dormant and only break the following year. And those buds which are infected will harbor the pathogen and show disease symptoms. So basically what we need to do is break this disease cycle by stopping the window of infection, stopping the inoculum from infecting buds, and protecting our buds. Basically, you have your diseased panicles, your available inoculum, and the window of infection of the buds, which is also the window of protection. So the strategy, therefore, is to remove the source of inoculum, which is the infected panicles, as soon as possible to remove those spores from infecting buds, and at the same time protect your available buds from infection with effective chemical sprays. I should just mention that there are many fungicides out there. In Israel, we've registered Prochlor as the most effective spray. Currently, we are working on new field experiments using new generation fungicides of results that are not yet available. I'd like now just to take you through a couple field chemical control experiments, just to mention one of them. In this experiment, we had 4 treatments. We have sanitation, which is removal of the panicles, as well as spray of a fungicide. Only sanitation, fungicide alone, and control, which is no sanitation and no fungicide. In this experiment, which was very complicated, as you can see, we measured cumulative yield tons per hectare over 4 years. This was the beginning of the experiment, basically the baseline in 2011. 1 year later, you can see already a difference between control and sanitation and fungicide, where the yield increased. And the following year, 2013, and finally, when the experiment was terminated, we found a significant difference in yield, a cumulative yield between the control and the combination of sanitation and fungicide. Another experiment, example of an experiment, we had many field experiments, but another one just shows you we have heat variety. And in this experiment, we only had 2 treatments, control, which included sanitation, and another treatment, spray, as well as sanitation in combination. This is a commercial orchard. We have rows of 65 trees per row. And again, the red is sanitation and spray, and the control or only sanitation. What I like to point out here, we have an infected mature orchard, which is releasing spores and releasing inoculum. So here we have our source of inoculum releasing spores into our experimental plot. And this is basically the summarization of 4 years of experiments. And here we counted only infected panicles per tree and not yield. But as you can see, the closest area or the closest rows to the infection source were the most infected. And as we were further afield from the infection source, so the infected panicles decreased. There was much less in the combined sprayed and sanitation, statistically significant, compared to the control, which is only sanitation alone. So to summarize here, we have our control and sanitation 2001, and over the years, you can see a significant difference between the control plus sanitation compared to the spray and sanitation, which had much less disease incidence. So just to summarize the management part of the talk, we need to protect our buds from infection, and this is the infection window. Now, in combination, we need to spray at the same time in order to protect the buds from infection and remove the inoculum to reduce disease. So you have a combination of removal of infected panicles, which are the source of inoculum, as well as spray to protect the bud from infection. So to point out, this is the way that the diseased panicles are removed in bags, taken out of the field, and usually burnt. Just to give you some pointers on what we plan to do in the future, on what we are doing as well, is to look at different species of Fusarium and do they cause similar disease symptoms to Fusarium mangifera, which we've been working on over the past 25 years. Devise methods for curing infected budwood. We know that budwood is the source of infection and can spread the disease within infected buds. So we're looking at dipping in different chemical methods, different chemical solutions, as well as heat treatments and combinations of them. We're looking again, as I mentioned, new generation fungicides for mango malformation disease field control. Currently, we have 6 new fungicides being assessed in field trials, and we're also screening germplasm and new cultivars for mango malformation disease resistance. I'd just like to acknowledge the funding sources for the work that's been done over these years. The Israeli Ministry of Agriculture, the Mango Growers Association in Israel, the USAID, and the FG program. Thank you very much for your attention. I'd like to take questions now.
Thank you very much for your presentation, Dr. Freeman. We do have some questions over here. So the first one is, do birds potentially spread the infection? We have white-crowned pigeons that come into the group to feed on ground cover and low shrub berries, and they love to land on the treetops. They return from the south in February and March when blossoming is going on.
That is certainly a potential source of spread of the disease, and that is only on condition that the birds land on infected panicles, they collect spores, and these birds arrive at buds which are potentially ready to absorb any spores which are available. So yes, that could be possible. I mentioned mites. We also can consider other insects such as ants, which may move spores from infected panicles to buds which are not diseased. So that's another point to make.
Okay, perfect. The next question is from Rocio, and she says, Dr. Freeman, hello from Nicaragua. We had the opportunity for you to visit their farm, and it was an excellent presentation. She has a question which says that they're performing a rejuvenation pruning that consists in lowering the trees to 3 meters high. They had the presence of Fusarium in these trees, but at 3 meters, they're clean of that presence, at least externally. Do you consider that these trees will be infested again, or is that a way to control the disease?
Well, as I mentioned previously in my talk, there's no systemic infection. However, any buds which can be infected and remain dormant underneath or below the pruning can be a potential source for future outbreak of the disease. So these buds should be monitored and the orchard checked on a regular basis to make sure that there's no rejuvenation of new buds, of new growth below where you pruned, where the fungus can remain dormant, and we know that it can remain dormant for many, many years. From my experience, up to 15, 20 years, the pathogen can be can survive within these quiescent buds.
So in that sense, it'd be better to do the pruning and a fungicide application, correct?
Well, the fungicide application would only protect buds and keep them clean. So if you have buds which are which need to be protected, yes, I would say pruning as well as spraying as a precaution. Yes.
Perfect. So the next question we have is from Martin, and it says, do you have any suggestions on products to treat the disease and application timing?
Products?
Yeah, like fungicide specific. Yeah.
Fungicides. The only fungicide which we've registered in Israel is prochlorades. There could be other strobilurins like Amistar. However, they need to be tested. Again, as I said, we have ongoing field experiment now with new fungicides from at least 4 different companies. So hopefully within the next year or two, we will be able to publish these results. As far as the second question goes, the spores are available as soon as mature panicles are present. So as soon as you see mature panicles, or even the beginning of malformed panicles, it doesn't need to be mature, remove them immediately. So you have to be very quick and on the ball and remove these panicles. What we, in the field experiment, we set up now, every 2 to 3 weeks, we remove these infected panicles. So it's removing infected panicles, eliminating the source of disease.
Okay. And I do want to mention real quick that Pulcherax is not permitted in the US and Europe. So we will look forward to the results from your fungicide trial.
Yeah, I know it hasn't. I mean, I work with other pathogens, and I work in strawberry, and prochlorades is the most preferable fungicide against Colototricum, and indeed, it's a problem. It was never registered in the US, and I think it's being phased out in Europe. I'm not sure about what's happening in South America.
We have another question from Martina. Could you repeat which of the countries have detected the disease?
Let me just
go to one of the first slides. Yeah.
Well, I'll just I'll read out those the countries where it was detected. Okay, originally from India, so India's highly affected. Australia, there have been numerous spottings. However, it's considered a quarantine organism because it's been eradicated every time there was a detection. Brazil, Central America. I don't know which countries. I know the Dominican Republic for sure because I visited there. Here it is. China, Egypt, Israel, Malaysia, Mexico, Oman, Pakistan, Puerto Rico, South Africa, Swaziland, Eswatini, Senegal, Spain, Sri Lanka, Sudan, Uganda, USA, and Venezuela, Nicaragua as well. I think omitted, Nicaragua. I'm not sure. I'm sure that there are other places, but I haven't been in contact with researchers from these other countries or seen anything in the literature besides the countries that I mentioned now.
Okay, so we have a question from Oscar, and it says, how does the witch's broom disease affect the fruit, apart in addition to reducing yield?
Witch's broom alone or in combination with mango malformation disease?
It says alone.
Alone, I'm not very familiar with the witch's broom. I know that there's a lot of leaf fall, of distorted stems, stunted growth. So I'm not aware of the how serious the witch's broom caused by the mango bud mite is concerned. I know that's the biggest problem as far as the mite problems in mango. I'm not sure of how serious of a pest it is and how the yield is affected, but for sure yield is affected.
Okay, so we are currently at the hour, so we're going to mark the end of our seminar. We will be posting the link, access to the recording on the National Mango Board's website, and we will send out an email with the corresponding information. We do look forward for your participation in future webinars. The following will be held on April 17th, which is on the existing technologies for fresh cut mango, and it will be from 3 to 4 p.m. And thank you so much, Dr. Freeman, for giving us your presentation, and thank you all for attending. Have a great day.
Thank you very much. Thanks.